Thank you very much, Marlies. Marlies is supporting us from the operational team in Biodiversa Plus, so thank her. Thank you very much, Marlies, for being here and helping us today. And very shortly, I will just like to, if I can pass my slide. Sorry, it's not working. Okay, now, very shortly, I would like for all of you, first of all, welcome you to the webinar, and then just say a very few words on Biodiversa Plus. So for those of you who are not familiar with Biodiversa Plus, it is a European biodiversity partnership that supports excellent research on biodiversity with an impact on policy and society. This is a very wide partnership. We do a lot of stuff, but here today we are representing the Work Package 2, who aims at promoting and supporting transnational biodiversity monitoring. The Work Package 2 is organized more or less like this. It will change now, but for a general overview, we work on identifying priorities and defining common indicators for biodiversity, and we work in harmonization and the use of monitoring data. And this webinar is organized by the Task of Novel Technologies and Approaches, who I am one of the coordinators of this task. And then we have some transversal activities like the biodiversity monitoring pilots that we are currently doing, and we will hear about one of them today. In the Task of Novel Technologies and Approaches, we designed a survey for novel technologies monitoring across the European partners, which are representative of the countries in Biodiversa Plus. We asked them what is the deployment state or their interest in deploying novel technologies for their biodiversity monitoring schemes. We asked them what targeted taxa and EBBs they were following and what challenges and constraints they faced in implementing novel technologies. All of that you can find it in this report. And then for capacity building, we are doing this series of webinars. If you're aware of this, if you are aware of this series, we did some previous webinars on image-based approaches on environmental DNA, bioacoustic, and sensor networks. But we have more webinars to come, so stay tuned. And yes, finally, for the important part of today, today we will have two wonderful speakers. First, we will have Maria Santos from University of Zurich, and also she's representing obligation today. Then we will have Sara Wiman. She's one of the coordinators of the Biodiversa Habitat pilot. She's working at SEPA, representing the government of Sweden. They will do their presentations now, and then we will leave some time for questions and debate. Just a few words on that. In this Zoom webinar, you have a question box. Please write whatever question you have for the speakers there. We will gather all the questions and we will ask the speakers at the end of their presentation. So please feel free to leave any comments or any questions, suggestions, or whatever you would like to say to the opponents. And now I will stop sharing my screen, and it's time for Maria's presentation. So welcome, Maria. Thank you very much for being here, Maria and Sara, and the support for Marlies. The floor is yours.
Thanks very much for the kind introduction, and good morning, good afternoon, or good evening, everyone. I'm Maria Santos. I'm a professor at the Remote Sensing Laboratories at the University of Zurich. So in the next several minutes, I will give you an overview of what have been tried out in terms of using remote sensing technologies for monitoring and understanding biodiversity. So where did we start? Well, we started already many decades ago by the first normalized difference vegetation index, NDVI, which you see here on the left-hand side, the original publication that showed that actually the reflectance spectra could be very useful to detect vegetation. And on the right-hand side, you see the first map of global biomes retrieved from NOAA's data from 1985 to 1987. Since then, a lot has happened. In the United States, the Earth observations from space is a document that review the first 50 years of scientific achievements using Earth observation. Some of them, of course, relate to this very powerful index and DVI, but much more has been learned since we have been able to monitor our planet using satellite technologies. What have we learned so far? We have now a wealth of Earth observation platforms. On the right-hand side, you see ESA, so the European Space Agency, and NASA Earth Science satellites that are currently in orbit and the ones upcoming in the close future. And through these different observation platforms, we have been able to monitor ecosystems, plant communities, and even some species. We have been able to also derive information that is very important in terms of understanding biogeochemical cycles, such as carbon, water, and nutrients. These are, for example, leaf mass area or leaf area index and other types of content of plant and their traits, and that help us understand how they actually work. We've also been able to monitor physiological processes. We have now instruments that are able to derive SIF, sun-induced fluorescence, and that help us understand photosynthesis, but also other instruments that enable us to assess transpiration and respiration, as well as stress to assess the health of our ecosystems. We have been able to derive a lot of plant and soil biophysical properties from chlorophyll to other contents such as water, cellulose, lignin, and so on and so forth. And also together with other types of non-optical sensors, and I'll talk about what I mean by that, we have been able to actually look at three-dimensional canopy properties such as height, area, vertical profiles, and gap structure. Where we started was to have data that enabled us to start to map how the ecosystems are changing. So here's on the left-hand side is, for example, the typical fishbone structure in the Amazon that is due to human expansion and changes in land cover types. What we see here is a recent image from RadarSat, also over the Congo Basin, that is trying to get us to the most recent types of observations, in this case, for water resource management. But land cover and ecosystem extent, although they have been traditionally in the most powerful use of remote sensing to date, it still has a number of challenges because it lacks still availability of value-added products, which include essential biodiversity variables. And the rest of my talk, I will focus on those. So what is it that we want to do? Well, to date, global assessments of biodiversity come from, for example, this predictive map of biodiversity intactness, which is based on in situ data that is combined in a powerful model relating biodiversity to land use changes to assess how much of the biodiversity that should naturally be present in a given location is still present due to land use and land cover changes. Moreover, the scientific community, au pair to what has been done in the climate community, decided to put forward an organizing framework that summarizes all the different dimensions of biodiversity that ecology, evolutionary biology, and so on and so forth have been examining for a long, long time. These are called essential biodiversity variables. And as I mentioned already, it's an organizing framework that divides biodiversity into its dimensions: genetic composition, species population, species traits, community composition, ecosystem structure, and ecosystem function. Moreover, essential biodiversity variables, as the graph on the right shows, they are fundamental to provide indicators of biodiversity and ecosystem services, for example, for the Convention of Biological Diversity, but also for many other types of uses. Some of these essential biodiversity variables can be directly measured or calculated, or even modeled using Earth observation. And the rest of the talk, I will focus on some of the progress that we have seen to date on how we actually make use of Earth observation of different types to understand and to measure biodiversity. One important aspect of what we want to do with these essential biodiversity variables is what we're doing within the context of obfuscation, is that we want to produce EBV enabling data cubes. What does that mean? It means that basically we want to combine data cubes of in situ observations that include citizen science to produce a data cube of observations, as well as remote sensing products, direct observations from space, airborne or experimental in situ observations, remote sensing products, and then derive eventually biodiversity products that will enable the development of these essential biodiversity variables. The road is still long, but we have been observing in the last decades a very, very fast development of what biodiversity measurements can we take by integrating in situ and remote sensing data. A couple of years ago, we wrote this perspective paper that we say that through the combination of in situ and remote sensing data, due to the different characteristics of remote sensing and due to the powerful information that comes from in situ data and the temporal domain, we are able to understand several types of biodiversity processes which are very fundamental. These include, not exhaustively, influences of evolutionary and biogeographic legacies, changing global distributions of plant functional traits, ecosystem resilience, past and present human modifications, and also how below and above ground processes are interlinked. We have a wealth of Earth observation platforms. We divide them in passive, which are optical, which depend on the incident radiation that comes from the sun and the bouncing back or the reflected radiation that is captured by the sensor at different heights. And then we have other aspects such as thermal, which is also a passive that we use the long wave radiation that is emitted to measure characteristics of our land surface. Moreover, we have two types of active remote sensing, LiDAR and SAR, where a beam of photons is bounced out of the different land surface characteristics, and we are able to retrieve three-dimensional structures. In terms of optical, for quite some time, we have been imagining imaging spectroscopy. What is imaging spectroscopy? Well, actually, it's also called hyperspectral remote sensing. And for the last 20 years or so, we have been developing the techniques, and now we're starting to have this type of information on satellite platforms. How does it work? Well, remote sensing started with single band information, as you see here, where we have reflectance over a given bandwidth within the electromagnetic spectrum. The different lines on the graph show the different land surface properties that we can measure and how their reflectance spectra could look like, depending on whether we're thinking about single band, multispectral like Landsat or Sentinel-2, and now with the advent of imaging spectroscopy or hyperspectral, that gives us the most detailed reflectance spectra that we can have from the different land surfaces. So why is this useful? Well, when we're thinking about biodiversity and the biosphere in general, imaging spectroscopy is very powerful. Why is that? Because the picture on the right tells us what should be the contribution of different characteristics of plants, such as chlorophyll A and B, carotenoids, anthocyanin, canopy water content, and so on and so forth, to the different wavelengths that we are able to measure with these spectrometers. The picture on the right and in the bottom is a representation in the RGB of what imaging spectroscopy can do, and it can have, when, for example, flown from an airborne platform, spatial resolutions down to 30 centimeters. And the picture on the right-hand side is actually showing you the data cube that comes with the image that is represented below. So we have between 150 and 240 spectral bands that we can use to retrieve information about the vegetation. So why is this important? Because different optical properties of plants, they are captured in the measured signal. This was proposed to be called an optical type, which basically comprises of information on vegetation structure, biochemistry and physiology, as well as phenology. Through this optical type, which is represented on the picture on the right-hand side, we have information in the visible about chlorophyll, carotenoids, and anthocyanins, about nitrogen, carbon, leaf mass area, and so on and so forth, in the near infrared and the shortwave infrared, as well as information on water content and other micronutrients, which are very important for functioning of plants and therefore of our ecosystems. So to come back to essential biodiversity variables, how have we been using this wealth of optical data, for example, to assess genetic composition? Well, recent advances using imaging spectroscopy have shown and have started to posit that we actually can start to understand the phylogenetic relationships between species, populations, and individuals by looking at the evolution of the spectral signature across the tree of life. This has been tested already in oaks, aspen, and beech, and some work coming here from the University of Zurich from FSC has shown that, for example, using a decadal time series of experimental site in the Legern temperate forest in Switzerland and showing a correlation between the spectral genetic similarity and different environmental variables, we can see that the spectra actually can correlate not only to the genetic diversity that basically expresses itself in the phenotype that we measure using the remote sensing, hyperspectral, and imaging spectroscopy data, but also it relates differently with different environmental variables. So basically, in summary, what the main point here is that we're starting to be able to say that different genotypes from different species, only a restricted number has been tested so far, look different under different stress conditions, and we can start to retrieve these when we have imaging spectroscopy data. Moreover, a set of colleagues have been looking into much broader scales. So they have looked into major phylogenetic clusters at global level that divide basically the Americas, Africa, and Southeast Asia in terms of their phylogenetic clusters. And what they are starting to examine is that the different traits that we observe today have strong relationships with these biogeographic and evolutionary legacies. Moving forward, species populations perhaps were the essential biodiversity variables that have had more applications from remote sensing. When I started, actually, I was tasked to track invasion, so map the presence of invasive plant species with imaging spectroscopy. We did this in many different locations in continental United States using an airborne sensor called HIMAP, but also another airborne sensor called Havris Classic. We had different compositions. So on the left-hand side, we had freshwater environments. We had water hyacinth. And then we moved into grasslands that were invaded and also ice plant as well as some vines. So we showed that to be able to track invasion with imaging spectroscopy, first we need to produce maps like the ones that I show here on the right and the top right, which show the distribution of the invasive species. And then we have to produce maps that show how the different species are actually functioning through time. So that's the maps that you see on the middle, that they show different seasons and how is the functioning of the different species over time. And then we can produce maps like the one on the bottom that shows us the persistence between 2004 and 2008 of the different invasive species. So therefore being able to track invasion with imaging spectroscopy. Why does it work? Because we need to think about certain characteristics of the data acquisition. First, we need to think about the timing of the phenological cycles. Most invasive species tend to be offset with the native species in terms of their phenological cycles, so we can time the data acquisition after the peak of the growing season or in timings that enable greater separation of the invasive plant species and the native species. We can use multiple data, particularly by combining data from different seasons. And especially now that we see a wealth of data emerging, we can start to do this much more reasonably. We have to think about the growth forms of these different species. And of course, the detection is best when the invasive and native species have very different growth forms. Furthermore, we would not be able to do this at this resolution, at 3-meter resolution, and in this case for submerged and floating aquatic species, if we did not have imaging spectroscopy. So putting all this data together, I placed it in a PCA axis, and you start to see that actually the different groups of invasive and native species, they separate quite well based on their characteristics. This is a good reality check to show that the grasses separate from aquatic macrophytes, and the graphs on the right-hand side also show us what we had expected. So if invasive species are really different from native, and therefore that's the reason why they succeed in certain ecosystems, they should have different spectral characteristics in certain wavelength domains. And that's what we see. On the green are the areas where the native species have higher spectral reflectance than the invasives, and in red, the vice versa. And I show this for 2 cases with grasses and macrophytes. For species populations, we're starting to see an emergence of many other technologies. I talked about active remote sensing, and here's some of the most recent examples of using LiDAR to start to understand the tree canopies, in this case, forest canopies, and being able to not only differentiate different species based on their architecture, but starting to understand species population. More is expected to happen in the very close future. Moreover, synthetic aperture radar has also been used. Here's an example to detect mangroves, because radar enabled certain penetration within the water, so much better technology to be able to detect mangroves even when they are submerged in some states. Moving forward, species traits have gained a lot of traction in remote sensing applications for biodiversity understanding and monitoring. So just to get us all on board, the definition of species traits comes together with functional diversity, and therefore the value and distribution of traits among organisms that simultaneously influence their individual fitness and ecosystem functioning. So as you can see, this is a very important aspect when we think about our ecosystems, and in particular when we're thinking about the dynamics of our ecosystems. There are basically 2 approaches. Well, we can add machine learning and artificial intelligence approaches here, but 2 approaches stand out to detect and quantify folio traits using remote sensing data. A typical one, statistical one, that relate, for example, vegetation indices of sorts with the characteristics of the plants. Or we can use partial least square regressions that enable us to predict trait values based on observation values. We can also use radiative transfer modeling and invert these to predict the different concentrations of certain types of folio traits of plants. A few years ago, the concept of spectronomics came about, and this was a concept that was put forward that would enable us to move to understand the canopy functional traits and its composition, as shown in the picture on the bottom. Ideally, we will be able to see a canopy like the picture thereabout, and several work have been demonstrating that folio traits can be determined from spectra using any of the techniques that I just mentioned for different types of tree canopies. And moreover, we can now start to place this in a geographical context, as the work that was led by the Greg Asner group in Peru, showing the different functional traits in geographical space. Moreover, more recently, set of papers have been trying to test the transferability of these types of models, and they used a wide variety of plant traits, 20 of them, comparing statistical models with convoluted neural network model from canopy spectra to predict which one of the models actually does better to transfer species traits models across different types of ecosystems. What they show is that not only some of the area-based traits show different types of correlations, but the models use different information from the spectra to predict the quantities that we are interested in. more work is expected to happen in the very close future, as this is a very active area of investigation. One work that we did also in the aquatic domain, so it's not just about terrestrial domain. We can also use this type of information for measuring, for example, submerged, floating, or emergent species. We looked into different types of vegetation indices that depicted chlorophyll and other pigments, leaf area, leaf water content, lignin and cellulose, and salinity. The pictures on the right show you some examples of how these trait maps look like. And the pictures on the middle with the yellow bars show the areas of the spectrum that we are using specifically for these types and groups of traits. So what we used this information to look into was to look into whether climate change was changing the trait distribution in this highly invaded estuary that I was talking about earlier. And we do see that indeed, by using data from the decade of the 2000s and again the decade of 2010s, we see that the traits really shape themselves in the PCA space, showing that the different plant communities are adapting their strategies or changing their trait spaces due to invasion and environmental stressors. This is also one of the aspects that colleagues are doing at the global level. What you see on the PCA graph on the left is the typical leaf spectral, leaf economic spectrum in PCA space, where we have leaf mass area, nitrogen content, and also leaf area and plant height. What you see on the right-hand side is what we can now retrieve with remote sensing of plant traits at a global level. And it's good to see that using these approaches that are still in development, we can basically retrieve the similar structure as we have based on in situ data. Moving forward to community composition, we see emergence of an approach that is based on the idea that emerged from spectronomics, that the spectral variability is an expression of spatial ecosystem heterogeneity and therefore related to plant diversity. So if that is really the case, then we can calculate diversity metrics that are very familiar to the biodiversity community, such as alpha diversity that relates to local richness, turnover in species composition, such as beta diversity, as well as gamma diversity, which is regional richness. The picture on the right shows the work by Jean-Baptiste Ferret in developing the method that enables us to calculate one of the first methods. There's more emerging now, and the R package that is now available to everyone to be able to calculate these different metrics of biodiversity. We have been working over the Amazon to calculate, in this case, the beta diversity, so a Sentinel-2 based turnover in spectral diversity over certain areas of the Amazon. Moreover, in terms of community composition, we're starting to see a lot of data and a lot of efforts coming from LiDAR data analysis. This is a picture where the authors try to predict community composition using LiDAR information, and they show that if we have information on shrub layer density, medium tree layer density, point amplitude, and relative biomass, we are able to derive good predictive models on the different compositions based on the three-dimensional structure of ecosystems. If you're interested in freshwater systems, well, we see the use of remote sensing a lot as input variables to, for example, species distribution models using eDNA or other organism in situ sampling to predict the community composition. Moreover, we see some examples that we can start to predict the different species presence within also freshwater environments. Moving to marine ecosystems, this is a picture of ocean color and the different mixing and also community composition that we can see at ocean surface. For the ocean, remote sensing of aquatic systems is a little bit more complex because water is a great absorber across the electromagnetic spectrum, so we can only use certain properties of the signal to measure aquatic surfaces. We base for the ocean on ocean color, which enable us to depict, for example, sea ice, sea surface temperature, sea surface height, salinity, chlorophyll A, and also chromophoric dissolved organic matter. Here's some examples of how these different properties can be mapped across different global platforms. The task on ocean science that is aiming at the Marine Biodiversity Observation Network has put forward this as a summary of the temporal and spatial scales at which we want to understand biodiversity in ocean surfaces, from the cell all the way to biomes, and what kind of ecosystem models can we derive for marine ecosystems that change across these different spatial scales of biodiversity processes. This is a process that is very much ongoing, but one aspect that has developed quite a bit at the moment is being able to monitor and map phytoplankton. Of course, we use this chlorophyll A content at the sea surface to be able to predict what kind of phytoplankton communities we can see in different points in time over different ocean extents. Moreover, other type of remote sensing, not light-based, but acoustic sounding, has been showing promises in terms of the deep ocean sounding and being able to depict biodiversity. This paper summarizes what we know in terms of being able and what are the advances in acoustic sounding to map biodiversity in the ocean layers. Moving forward, we have ecosystem structure. Here is where LiDAR comes about. So here's an example of a three-dimensional structure using airborne LiDAR, but we also have terrestrial laser scanners that enable us to get a more complete picture of how the LiDAR looks like. Here's another example of a drone that has a LiDAR sensor mounted in it, and this is a video from DJI, which is a drone company that offers LiDAR-equipped UAVs, and so it shows you what kind of properties and how the properties can be assessed when using this type of imagery. Moreover, we can also do it in estuarine and aquatic systems you see here. And of course, as you can imagine, water surfaces do not have a lot of penetration of LiDAR signal, but at least we can see some sedimentation and distribution of characteristics that are important for freshwater, and also interface between fresh and saline water and biodiversity. In terms of ecosystem structure, we have seen the first ecosystem LiDAR in space, JEDI. And JEDI has basically three science questions. What is the current state of Earth's forest structure? What will forest dynamics look like in the future? And how does forest structure affect habitat quality and biodiversity? You can see the mission overview description in the website that I've pasted here. But here's one of the first analysis that shows over the continental United States a distribution in RGB. So in red, we have maximum height. In green, we see foliage height diversity, and in blue, we see plant area index. So areas that are in red are dominated by maximum height, and then the combinations thereafter show the different representations of what we can actually do using this ecosystem LiDAR from space. Other emerging topics are what kind of footprint do we need from radar to be able to capture different types of ecosystem structure. And this is what this exercise here shows. They tested different grain sizes in terms of the footprint and the different retrievals from terrestrial laser scanner in this case to represent different characteristics of an ecosystem that might be of interest to represent its ecosystem structure, and therefore, for example, the biodiversity that depend on openings or that is distributed along the three-dimensional characteristics of the canopy. Moreover, we're starting to see the emergence of synthetic aperture radar also for biodiversity applications. Here you see how the different polarizations of different radars can differentiate human-made infrastructure as well as forest, and specifically and very importantly, water content in the canopy. We have been testing some data from DLR Airborne FSAR. We have a project funded by European Space Agency where over Lagern, the same site that I've showed you before, we can now depict vegetation. So in blue and dark violet, you see bare soils or very short vegetation such as grasslands. And then in green, we see leaves or branches larger or higher than 15 centimeters to 30 centimeters. In other colors, we see the artificial objects such as houses, power lines, fences, street furniture, signs, traffic lights, and so on and so forth. Moreover, when we're thinking about ecosystem structure, it's not just about structure in the vertical domain, but also in the horizontal domain. Here we come back to spatial structure of ecosystems, and we've been developing a Bayesian approach to look into not only the typical land use features in the landscape, but what we call hidden features in the landscape. Through this model, we are able to detect, for example, patterns within continuous fields variables. These are some of the examples of what this might look like, but we tried it for example, continuous field properties such as normalized difference vegetation index, land surface temperature, latent heat, land cover, and then digital elevation models. So through these, we are able to depict hidden features in the landscape that might matter for biodiversity in terms of habitat structure, but also in terms of habitat quality. To finalize, ecosystem function. Ecosystems are dynamic. Many of these measurements, although we're starting to see a lot of temporal resolution in many of these applications, we need to understand how ecosystems function over time. This is through measuring, for example, phenology of ecosystems. We see that phenology has had a very long history of interest for many types of applications. We've had planting, pest control, pruning, all depending on our understanding of phenology periods, in this case for agriculture, but also for natural ecosystems. Phenology emerged as an independent discipline sometime around the 1990s, and nowadays we have cutting-edge monitoring technologies from satellite and monitoring techniques. We have been using the sensor that we have here at the University of Zurich and the legacy sensor, APEX, to assess how can we do actually phenology over certain types of ecosystems. One important aspect is if you're trying to map, for example, grasslands in Switzerland, is that we have to think about pre-processing steps when we are collecting this imagery, not only in terms of atmospheric compensation, which is very variable, the distance between the ground and the aircraft when you're flying over very rugged terrain, but also we have to consider topography correction. What we see by using a temporal data set by the Airborne Prism Experiment sensor of the Swiss National Park over at least 8 repeated times, we're starting to examine what is the effect of these types of corrections and how can we actually choose the different corrections when we're thinking about, for example, mapping biomass over time. We see that, for example, corrections have an influence, but we also see that models are relatively stable if we use at least mean-earth corrections. Moreover, we might actually benefit from the temporal domain and the dense time series by using machine learning artificial intelligence technique that is emerging now, which is called guided super resolution. In summary, what this technique does is to benefit from the different repeats of acquisition and the slight geographical misalignment between the different pixels to create a super resolution. What we show in this paper is that if we use a scaling up approach, these are the results. If we use regression model, these are the results. But if we combine those by guiding the super resolution extraction via high resolution image, we produce much better results in comparison to what we measure on the ground. However, this is not true across all of the above ground biomass values, and the models do better at lower biomass values than at higher biomass values.
Maria, do you have 5 more minutes?
Okay, perfect. Thank you. So in terms of ecosystem function, we also see the new launched biomass satellite by ESA. The first data was released in July this year, and we will be able to see this coming into the future. Finally, thermal data is starting to be used to assess the thermal conditions of our ecosystems. Trishna, which is a thermal infrared imaging satellite for high-resolution natural resource assessment, will be launched, and now we also have information from EcoStress to assess function in terms of the thermal domain as well. So to conclude, we are going very far when it comes to monitoring and understanding biodiversity. We've seen different approaches with different technologies with variable accuracy and results. And we will see in the future, this is an example of the Copernicus Sentinel expansion missions that will come into the future. So from above, from space, we will have a lot of data coming in, but we have some needs as well. We need a lot of in situ and in silico data. We need to be able to do better in terms of our uncertainty budgets and to be able to provide robust estimates and also protocols for what is acceptable for these biodiversity metrics. We need better super resolution and multimodal integration. When we're talking about biodiversity, it's about diversity of systems. So central tendency methods might not be the most adequate, and we may be missing out on the fundamentals of biodiversity, which are focused on rarity and uniqueness. So new methods are needed to capture what is new and unique. And then, of course, we need full spectrum contributions, and we need to identify which dimensions are most important for these rare and unique characteristics of biodiversity. And finally, we need to move beyond pattern description to understand causal links between the different drivers of change. In terms of in situ data, we have very limited geographical coverage. The image on the right top show the FluxNet sites where we actually have flux towers that measure ecosystem function, and the bottom graph show in black the sample pixels for the TRI database that gives us information on plant traits, and areas in green show us how areas are intact in terms of biodiversity, and basically we see that we are missing in the areas, we are missing observations in the areas holding most biodiversity. Moreover, we need to be aware of uncertainty budgets. We are reviewing and providing information of how do we actually design these uncertainty budgets and how we calculate it over spatial surfaces. So if we think about the processing chain for remote sensing for biodiversity products, we see that uncertainties, they accumulate. At the radiance level, we have between 3 and 8%. Topographic and atmospheric correction more than 20%, surface reflectance more than 5%, and only considering field surveys we have more than 10%, and we don't have even the calculation of the uncertainty when we go through this step, for example, to calculate spectral and functional diversity or species distribution models, or include information from remote sensing onto vegetation models. We're starting to do that. So here's an example of an uncertainty estimate for true wavelengths from the DESIS sensor. And here's how this propagates into, for example, the chlorophyll to carotenoid index or the spectral diversity. The two images on the top right, they show the index calculations, and the two ones on the bottom show the uncertainty around those estimates. Moreover, and just to conclude, we can monitor a lot. We are in this phase of the observation. It's super important to detect and observe, and we've seen a dramatic increase on what the capabilities are for measuring many of these domains of biodiversity. But we need to understand theory, develop good causal models, estimate, detect, and attribute what are the drivers of change. So with that, I wanted to conclude by saying, well, biodiversity is about generating innovations. Here is a chlorophyll photosynthesizing sea slug. Understanding biodiversity is challenging. I don't need to tell you this because I'm aware that you all know that. It requires a lot of creative and novel ways to use Earth observation beyond measures of ecosystem properties. But we see a wealth of new data and technologies as well as analytical platforms to starting to enable us to understand biodiversity. So with that, thank you very much for your attention.
Thank you very much, Maria. That was a great presentation. We got some feedback, very positive feedback in the chat. And then the floor is now open for Sara whenever you're ready.
Thank you very much. That was quite a lot, Maria. Thank you very much. Interesting. So, okay, I share my screen.
Yes, please. We've got some questions that we will begin answering later after Sara's presentation. If there are some short ones, Maria, that you want to answer, you have the possibility to do that in the question box as well. Sara, we can see now your slides, so please.
Yeah, thank you very much. Okay, I will cover quite a much smaller area because I will present to you some examples from the work we've been doing in the Habitat pilot within the Biodiversa Plus. It has been going on for 2 years almost. And right now we are finalizing the results and validation and writing the final report, which is due end of this year. So what we are trying to do is to evaluate the capacity of remote sensing to use for an objective and comparable way of mapping habitats and monitoring the condition of habitats in grassland and wetland over Europe, and to support the development of harmonized monitoring protocols in the EU. And I will talk a little bit, I should say, also who are involved. All the green countries have been very involved in the whole project, and the orange here have been involved in the first phase. And the first phase, starting this project, was to make a comprehensive review of existing methods, existing data. How do we do the reporting and mapping in different countries? So we collected all information we could find and listed them as potential tools for a more European-wide use to make it more comparable. So here are some examples of the different methods we described to ourselves and discussed internally. Here is a group from the first workshop in Italy, where we went out into the field and got some valuable insights and got to know each other better. So the input data, after the review, we choose a number of focus areas. What would we dig into and try to validate in the different countries and try to test it in different countries and see, is it the same? Is it comparable? Can we use this for an upscaling? Use it more widely. I just mentioned some of the input data. It's not only satellite data, even if, as Maria said, there are huge numbers of different satellites up in the sky right now in the space, and it has been available since from the Landsat series starting in 1970s. Then for the historical reasons, it's very important to have this old data, which is not very detailed, but it's still there, showing what has happened in the longer time frame. So we also use drone imagery, LIDAR data, climate information, water tables, existing maps, of course, habitat maps, if there are any, and field observations area photos, but mostly Sentinel-1 and 2. There's an image of how many satellites are there launched around the world per decade, quite a lot. And the methods we have been using and focusing on is mostly time series, which is extremely important for monitoring changes and variations within seasons and between seasons, between years. For example, in looking into inundation. Some machine learning for classification of habitats, and also super resolution, which is a AI models trained for creating high resolution data from, for example, Sentinel-2. Segmentation, image segmentation to delineate areas that are homogeneous but considered to be one habitat is extremely difficult, but there is a method we have tried and evaluated. And also looking into validation of existing habitat mappings and other UNIS classification, for example, but mostly EU Grassland Watch, to see, is it usable? How good is it for our purposes? So this is how we divided the subtasks. We choose some, and then we had some partners. All the partners could choose to work with different subtasks, depending on, for example, all the countries didn't have a EU Grassland Watch map yet, so they couldn't participate. And here's a typical botanical image from our last workshop in October in Diest in Flanders, looking into some wetlands. And wetlands, that was one major that many partners participated in, and the aim is to produce robust indicator time series from data. And we tried different models, for example, Jössela model, which is a new one developed by a partner. You can see it down to the right here. It's a decision tree using spectral bands and some indices as well to define, is it dry or wet, or what is it? And then you can get, you have a production line out from that produce a long series over the years. And to the left is another one tried out also by several partners, STR, and showing a little bit not as wet as the Jössela method. But the idea here is to see which the importance of seeing what areas are permanently or often very longly inundated very long time, which is bad for the biodiversity. It should be not as long. So you can monitor by using this time series and get the point of time when something happened. Of course, there are outliers, and it's cloudy and it's difficult to get pure pixels everywhere. So here you can see the different indices used within the two different comparisons. Let's see here now. Okay, so here is an area in Finland where you have a permanent water bodies and seasonally inundated grasslands. So you can see the permanent water in green below in the bottom and dry areas in yellow. And you can also see how they are reflected in the overview of the area. And the light green is seasonally inundated. And it's important to define how long time they are inundated, because if they are too frequently or too long, you will lose biodiversity. Another area, this is in the east. And so the problem of satellite data, as we use Sentinel-2 for this, you don't always get good data, because is it clear weather? Of course, you get fine pixels everywhere, but is it cloudy? You have cloud shadows, you have atmospheric disturbances of any kind. So here is an image of how complete the data is. The darker, the more pixels per month. So this is already from year 2020 to now, until now. Some months are empty. You don't get any pixels at all. You cannot get an image covering this area, even if the satellite is the visit time is like 5 days, or rather 2-3 days, actually, in Northern Europe at least. But is it cloudy as it was? Then that's a problem. So then sometimes you have to fill in data to get the accurate understanding of the curves and variation over a season. I shouldn't dig into this too much, but we tried a lot of different indices to see which are reflecting what we see on the ground the best, and the combination. So this is the development of finding out how to treat the data that we have when we have good pixels, good data. And referring to the spectral curves Maria showed, this is how water, for example, is reflected in the SWIR band, which is the long wave infrared. The more water on the ground, the less it reflects. So very low values in the SWIR band shows that there are a lot of water. And using this knowledge, you can combine and develop specific indices and combinations to really see what you are after. But this varies. I think it's important to know that different areas, they behave differently, and you need to take into account the structure. For example, you have the Apamyres in Finland with where is kind of a mire with water in between in long it's a pattern that is very peculiar, actually. So when you have missed some observations, it was bad data, no satellite data, maybe you need to interpolate. So here we just tried out. You can interpolate and try to fill in the gaps, but still you have perhaps some of these points are outliers and not good data. I hurry on with this one. So what we found out in the project was that it worked quite well for detecting, separating open water from drier surfaces, and also get this long-term seasonal variations. Limitations would be small water bodies, that it's the resolution of the satellite, 10 meters, is too low to map them properly, because then you get something we call mixels, that is a mix of anything. You can have water which is reflecting low in all the spectral bands, and you can just outside, maybe there is vegetation, healthy vegetation reflecting a lot, especially in the green and infrared bands. So that was a limitation. Some variation between different countries and depending on what is growing there. For example, reed is a real problem because you cannot see the water because the vegetation is covering it. And readiness for monitoring. So it's quite a easy way of handling data, but it's not actually, you cannot see the species clearly. You can see the second, information about an area that is could be interpreted as a valuable habitat, depending on how it behaves in this observed arithmetics. So we would really like to continue with this and scale up it in the future. Because of the problems with small water bodies, and then we tried out the super resolution, creating there is a model. If you see up in the right corner here, there's a satellite image, Sentinel-2, which you run through a model that is trained on orthophotos or very high resolution satellite data, and the model itself contains then all the information used for transforming one satellite image to a very high resolution satellite image. It's the same image. You don't use anything else. So this is a little bit of it's quite amazing actually, because if you use it in a city, you can see so properly that you can see all the houses, the roads, very, very detailed. But in this case, we tried two different ones that they had some difficulties reflecting the spectral information, that it differed quite a lot, but and it invented details that were not there. For example, the SATLAS method that was trained on American data, it invented that, okay, here's a open grassland, but it invented that it would be something much more detailed. And we could see in the Oppenmeyers, it made sort of American suburb area with plots and everything, even if it was completely flat area with only wetland. So quite tricky, but we believe in this for future development. And also the SATLAS model was a bit tricky because it uses 8 input sentinel images, so not only one point of time. So it integrates, maybe something like Maria's model that she showed.
Sara, if you could do 5 more minutes to
Yes.
leave some time for questions.
Yeah.
Thank you very much.
Okay. You could, to enhance the the spectral information, it's possible to histogram match. And first you got quite, the colors didn't look the same. Also, one of the methods was only RGB, so it's quite difficult when you really need the sweet band and near infrared. However, we made an evaluation, and I talked about the mixels with the 10 meters mixels when you have at the border, and you get this confused pixels that they don't know, is it a bit of water, a bit of vegetation, and they are in the middle blue here. So using super resolution reduced that amount significantly, so that you get more pure pixels and can analyze it more detailed. But the information in the data doesn't necessarily increase, just the outline of the phenomena. About the same in confusion matrices, the same result on Sentinel-2 original data as in the S2GR3 super resolution image. Here are some other examples, trying a well-known method, Lefebvre, comparing to Jössila method that we used in this. They are a little bit different depending on how you threshold and how you map. And then going out in the field, looking into this, you can see that it's not very easy, not with any method, to exactly, for example, it looks like you have very dry areas in the middle of a very wet area in both cases here. And then we come into this validation part, which we consider is extremely important for validating the methods itself, comparable for upscaling purposes in the future. And it's quite difficult, because even if you say you do field validation, it could be so extremely variating what you see and how you interpret it. Here are some image examples with high reads in Czechia, and it looked dry on the surface, but then you sink down in the water. But we used quite a lot of different ways of collecting reference data. Visual evaluation based on aerial images, for example, it's a very common one. Point sampling. So we did the validation of EU Grassland Watch. This will finalize the session then. Performed by all partners possible. It wasn't available everywhere. And we created like a tool for how to, and a solution for how to stratify the points and how to analyze this in the same way in all the different countries. So we made a little tool and we discussed how to interpret among the partners. And we had the workflow of how to first stratify the points and then to go into the tool, which jumped around the points, and you can decide yes or no. What is this? Is it correctly classified or not? And then it automatically compiles an assessment report. This could be used for any other evaluation as well when you use point evaluation. Okay. We also tried out segmentation, which is to delineate areas, homogeneous areas. And so then I finalize. Thank you very much. We are finalizing the project now, but you're welcome to contact us. And I can see here that one has retired, Ola. We are this project has been co-led by Finland and Sweden during these 2 years. Thank you very much.
Thank you very much, Sara.
Question?
Yeah, we will stop the recording now before going to the question.